Okay, so third topic in B1, infection and response, and mostly about communicable diseases, so the ones that we can catch and how our body fights against those. And then those second set of bits are really just for biology only. So, moving on, first thing, when asked the question of what causes disease, a lot of people say bacteria, or some people say viruses. Actually, the correct term up here at the top, must know word, pathogen. And pathogen is an organism that causes a disease. They are microorganisms, and yes, there are 4 possible types of pathogen. So when you're looking at this, just change colour pen. When you're looking at it, you've got 4 types of pathogens, and those are bacteria, viruses, fungi, and protists. So, bacteria we've done in cells. They are similar in some ways to plant and animal cells. They have a cell wall, they have a cell membrane, they are prokaryotes in their own domain. They do not have a nucleus, much smaller than normal cells. Viruses are more just a protein coat, not even made of cells, a protein coat with some DNA or RNA inside. Fungi, such as athlete's foot, etc., whole completely different kingdom. And protists are single-celled eukaryotes. So these ones do have a nucleus. They're bigger than bacteria, and an example there would be Plasmodium, the protist that causes malaria. So, moving on. How can you be infected by a pathogen? Well, here's your variety of methods. You could have it by using a syringe that isn't clean, so therefore you're injecting it into your body, getting it into your blood. So some sexual diseases, sexually transmitted infections, etc. Some HIV virus, etc. is transmitted in this way. It needs blood-to-blood contact. So here we've got people crowded, and we know about social distancing now. So transmitted through air and coughs and sneezes. Contact by athlete's foot down here is usually picked up by direct contact. So you going into a changing room and standing somewhere else, somewhere where somebody with athlete's foot has had, you could catch it on your skin. We can catch it from water. The water can be infected, and that's in our country that's not so bad, but in countries where there's not so much water purification and having contaminated water, etc. So we can have it spread by water, we can have it spread by air, and we can have it by direct contact where you actually need to be touching the organism itself. And then you've got the needles, etc. So, okay, so for each of those groups of pathogens, you're expected to know some of the main examples. So here we have our viral diseases. What are the viral diseases that you need to know and memorise? You've got measles, which is a red skin rash, can be fatal if complications arise. So if you were ill from something else, or you didn't have a very good immune system, etc., or you were very weak and very young, measles can be quite dangerous for very young babies, etc. How is it spread? Measles is spread by inhaling droplets from other people who have it, coughing and sneezing. How do we control it? Well, we vaccinate young children. So most people have measles, mumps and rubella injections. It's one of the first, the MMR injection vaccination. HIV, initially this human immunovirus is flu-like. It's spread by sexual contact or exchange of body fluids, such as we said on that last slide, sharing needles. So blood-to-blood contact or fluid-to-fluid contact. It was spread around amongst drugs users and by people having sex without protection like condoms, etc. Unless it's controlled with drugs, and obviously now we have antiretroviral drugs to actually fight it, it will attack your immune cells. So HIV itself doesn't necessarily cause you that much harm, but your immune system can no longer deal with other infections or cancers. So other infections and cancers are not fought off by your immune system because your immune system has been attacked by the HIV virus. And then in plants, tobacco mosaic virus, and first found in tobacco plants, etc. It's very common there, but it does affect other plants as well. It causes a mosaic discoloration pattern on the leaves. That means that it's destroying some sections of the leaf that have got the chlorophyll, so it can't do photosynthesis as much, and that means that if you can't do photosynthesis, you're going to have poor growth. So less light is being used, less food is being made, you're not going to grow as much as a plant. How do you control it? Well, we can take away the affected leaves so it doesn't spread around the plant. Quite often we burn the crops that have it to stop it spreading from one field to another to another. So usually we'd get rid of that whole crop and getting rid of the pests and stuff that can carry it from one to another. Okay, now bacterial. So one that a lot of people will have met or the parents will have had, food poisoning. It's a bacteria that you take in through your digestive system. It's ingested in your food or in conditions where the food has been prepared in unhygienic conditions. That makes you feel very ill. You get stomach cramps, diarrhea, vomiting, and poultry are vaccinated against salmonella to try and control the spread of it. So a common place you'd find it is in eggs if they're not cooked properly, in chickens, etc. But poultry vaccinate against it to stop them spreading it around. Gonorrhea. Sexually transmitted disease, a bacteria, so from fluid to fluid again, rather than through the food. And always a great image to think about, thick yellow or green discharge from the penis or the vagina. Pain when urinating. Now, the good thing about bacterial diseases is they can usually be controlled or fought off by antibiotics. How would you protect yourself from getting gonorrhea in the first place? Something like a condom. Fungal diseases. Just one to learn, and it's in plants, a common fungal disease. Obviously knowing that athlete's foot is a fungal disease as well helps, but rose black spot is exactly what it says. Black spots on the leaves. Purple or black spots, you can't any longer do photosynthesis through those black areas of the leaf, and therefore your growth is reduced. We wouldn't want our crops to be growing smaller or food crops to have this, etc. The fungal spores, this rose black spot, are spread by the wind. And again, we can use a fungicide here. A fungicide is something that will kill the fungus. Again, what we might do is if we've got a crop infected with rose black spot, we could decide to harvest that crop, burn the whole lot down, get rid of it so it's not spreading around. It might be a cheaper alternative to take a risk doing that rather than just treating some of it and hoping it doesn't spread. And then we have the protist disease. So malaria is caused by a protist, and the protist is actually the vector. The vector for malaria is the mosquito. So mosquitoes don't actually cause malaria. What mosquitoes do is when they, and there's a little mosquito down the bottom here, when the mosquito bites you and then he goes off and bites somebody else and then he goes off and bites somebody else, the mosquito can actually carry that protist between one person and another, hence spreading the disease around. So controlling the number of mosquitoes in an area and stopping them breeding, etc., preventing down the bottom here, stopping the spread of mosquitoes and the breeding, using mosquito nets when you're sleeping to avoid being bitten, etc. What that means is you can protect yourself from being bitten by the mosquitoes and therefore hopefully not get malaria. So inside your blood you get a recurrent fever, and that can make your body very weak, and that fever can last for a long time. Then you can get better, and then it can come back again and again as the malaria and your body fight against each other. If you're weak, if you're malnourished, in a lot of countries where malaria is a big problem, in the warmer sort of tropical countries, if malaria is a problem and food shortage is a problem as well, if you're already weak, etc., malaria may well be fatal. So there's a summary of all the ones that you're expected to know, and next to it, here are the symptoms. How is it transmitted? And then I meant to explain this a minute ago. A vector is how it's carried. So for example, rats are seen as disease carriers, so rats would be a vector of some diseases. So rats would have the diseases and be spreading it from place to place. The rat itself isn't the disease, so here the mosquito is the vector. The little protist himself causing malaria, Plasmodium, that protist is being carried by the mosquito, so the mosquito is the vector. It's the method of carrying that. And then the others are just a summary of what we've done so far. So moving on, I'm going to flick through the next few slides quite quickly, because you can pause it and answer questions, and then you can pause it and mark your own as well. You don't need me to talk you through it. Okay, and now the answers exactly the same. Pause as you need to. Okay, how do we defend against these diseases, and what can we do about it? Well, first thing is, as we were saying, our nose. So the nose and our breathing system. So for diseases that are transmitted by air, we have defences. So we've got hairs in our nose. We've got mucus lining the inside of our nose and our windpipe, our trachea and bronchus. We have cilia, these little hairs on the cells that move the mucus up. So basically, through our nose, and then over here, as you go down the trachea and through the bronchus, etc., the idea is that our mucus will trap all the dirt and germs, the pathogens, and the cilia moves them up, and then they're swallowed, and then they end up in the stomach. So they don't actually get to infect our lungs because we stop them getting in. Now, one of the other ways was that we can get diseases like salmonella from our food. And then we've also got the fact here we're swallowing these ones that we breathed in. So they end up going into our stomach. One benefit of the stomach being acidic, because remember it's acidic for digestion, but being acidic, that pH, strong acid, it will kill most pathogens. Now salmonella is one of the few that actually can survive in there and cause infections in our intestines. Then again, with the air and waterborne pathogens, we have our skin. Our skin has dead cells on the outside here, those dead layers, so they don't get infected so much. The skin's quite hard to penetrate. It's much more risky when you've got cuts in your skin, etc. Sometimes germs find their way down here into the pores in your skin, and they might cause an infection or a spot and a swelling in the skin. But actually, it's quite hard. Our skin is airtight, our skin is waterproof, therefore our skin is protecting us from these pathogens from the air and water. Okay, so pathogens getting into the body. Then it's the job of our immune system to take over and fight the nasty pathogens off. So, why does the immune system recognise a pathogen? Pathogens are different. The white blood cells can tell which cells are yours and which cells are not yours. Their job then is to attack anything that isn't yours. That's why after maybe a transplant operation, we can reject organs, because your body recognises it's different than your normal cells. So, there's three ways white blood cells can defend the body. And the first group are phagocytes. Phagocytes are the white blood cells that actually, there's a little germy, here's a white blood cell, here's a white blood cell, and it surrounded the germy, and then it digests it and destroys it. Gone. Germ gone, white blood cell, carry on patrolling, find more germs or pathogens. So it engulfs it, that means surround and digest it. Second thing a white blood cell can do, the lymphocytes, they recognise antigens. They recognise the proteins on the outside of the pathogen, and they make antibodies. So you can think of these as missiles. So they basically make antibodies. The antibodies then destroy the pathogen. So this time they don't have to actually find the pathogen themselves, but they recognise its presence in the body, they make antibodies, they attack the pathogens. That can take time, so you may get an infection and then you get better as your white blood cells fight it off. Now, the second time that you meet the same antigens, your body might be able to react a lot faster. So your white blood cells can react faster sometimes and make antibodies much faster when they've met that pathogen before. That means we gain immunity, so we fight it off and react to it much faster. So something like chickenpox. Chickenpox, the first time you get it, the number of bugs goes up and up and up and up and up and up really quickly as we learn to fight it. Then we kill it off and the numbers go down. The second time your body gets chickenpox, what should happen is the numbers start to go up, and then your body quickly responds and fights it, and you don't even know, and you don't even get the symptoms of chickenpox. So then we've developed immunity to it. And then the third thing our white blood cells can do down here at the bottom, one of the things that makes you feel ill is the fact that the bacteria produce toxins, and so the job of the white blood cells is to produce antitoxins. And that's how a lot of medicines work. So something like night nurse, etc., they contain antitoxins to make you feel better and get rid of the toxins that are in your body. So white blood cells can make antitoxins to get rid of that. Okay, so white blood cells, recognising a pathogen here, producing antibodies destroying the pathogen. Now, in order to give somebody a vaccination, in order to give somebody immunity and help the immune system, a vaccine contains dead or inactive form. So when you were given the measles, mumps and rubella vaccination as a very small child, what was injected into you was dead, weak versions of the measles, mumps and rubella. Your body then thought, oh, I'll learn how to fight these. Your body then remembers how to fight them, and then it can do that quick response in the future. So when you catch measles, as the number of measles are going up, you quickly fight it off. So you don't actually develop the symptoms of the rash and the fever, etc. So we call that immune to it. It doesn't mean you don't catch it, it just doesn't last in your body because your body can fight it off really quickly. So you've been given immunity to it, and that's why we give vaccinations, especially to children. This just goes through exactly the same thing. Antibodies released much faster the second time round, pathogens destroyed faster, unlikely to suffer the symptoms. So therefore you've developed immunity. Antibiotics can fight bacterial infections. Now, antibiotics are specific. So some antibiotics will kill off some bacteria, others will kill off others. So we need a range of different antibiotics. Antibiotics cannot be used for viruses. So viral infections not affected by antibiotics. Antibiotics are for the bacteria. Penicillin would be the one antibiotic you'd be expected to know the name of. Some bacteria over time have mutated and they've actually become antibiotic resistant. So there are now bacteria who, I'll draw their cell wall on as well to keep it cut, and there's their bits of DNA and a plasmid. This bacteria here now, this is now immune to penicillin. It might be immune to amoxicillin. Therefore, eventually, if the bacteria keep developing immunity to antibiotics, we might run out of antibiotics. So one important avenue of medicine is continually finding new antibiotics and using them properly. So this is just talking that through. Bacteria can mutate, they can become resistant, and that's an increasing area of concern. I'm just going to move on quickly. And questions? Pause and answer. Pause and answer. And once you've done those, did you get the answers right? Which areas might you need to revise again? You can go back in the video or look elsewhere for research. Again, you can go back a bit and look elsewhere. And now, discovery of drugs and development of drugs. So, many years ago, in the olden days, a herbalist or somebody would give you plant extracts, maybe certain herbs for certain conditions. And actually, aspirin, which is a painkiller, around much longer than paracetamol or ibuprofen, it's a painkiller, it's an anti-inflammatory drug, so if you've got a sprain or inflammation, willow bark. So in the olden days, people would be chewing willow bark and it would help make the pain go away, etc. Not understanding how it worked or how it worked on the nervous system, but now aspirin can be got from willow bark and we can sell it as tablets and stuff. Foxglove plants have an extract of digitalis, which helps with heart regulation and heart health. Mold, penicillium, is a mold that actually naturally it produces a chemical that kills bacteria, discovered by Alexander Fleming. So actually what we've done is we've purified it, we've taken this drug penicillin out of the fungus, and now we actually take that medicine ourselves. So we take penicillin as an antibiotic to kill off bacteria infections in our body. How do we make new drugs? Well, there's a whole testing process. So, 3 things here for testing drugs for. Efficacy, make sure you know that word. Does the drug actually work? Toxicity, what's the point in having a drug that has massive side effects and makes you ill in a different way? So is it poisonous? And then the dose. What is a safe dose of that drug that doesn't cause side effects or damage elsewhere? And so we start off with preclinical tests. And these are tested before any trials on humans are allowed. So, preclinical tests are, we experiment with cells, we experiment with tissues in a lab and look at how the drugs react to tissues, do they kill cells, are they harmless, etc. And then we try simple little animals. Once we get to the clinical trial stage, there's 4 stages. Here, first thing is, healthy volunteers, try some of the drug, is it safe? Are there any side effects? So the first thing is, does that drug actually make you ill or affect your health? Second thing, some patients with a condition try the drug at a low dose. Does it seem to have any effect? Does it seem to help them improve? Then, if that looks like it's the case, so we now know it's not poisonous, doesn't cause any problems really, or ones that we're quite happy to have, minor problems, it does seem to help with that condition. Now let's do larger number of patients taking the drug and taking different doses of it to work out what's the dose that has the best effect. So you might go with 5 milligram of the drug for this person, 10 milligram for this person, 15 milligram for this person. This person, it only mildly helps. This person, it helps loads. This person, it helps a little bit more than 10. Actually, let's go with a 10 because it seems to help loads, and actually when you get to 15 it causes migraines all the time. So we work out the right dose, and then we do, to see how effective it is, the efficacy of the drug, we do a double blind trial. So what we do is we do a trial where some people are a placebo. Some people are actually getting the medicine, and some people are getting a pretend version of the medicine. And we look at the differences between the 2 groups. So, people are chosen randomly for this, each person randomly. They won't know who is receiving the real drug, and they won't know who's receiving a placebo until the end. So that means we can actually measure what effect the drug has. Is it just taking a tablet that makes you feel like you're getting better, or is actually the tablet is the drug actually having an effect in treating the disease?